Huntington's disease is inherited by autosomal dominance. This means that if mum or dad has a mutant gene, there is a 50% chance that their children will get it. On chromosome number 4, the HTT gene codes for the Huntington protein. The exact function of this protein is unknown, but it is thought to be related to the functions of neurons in the basal ganglia in the brain. In the HTT gene, the trinucleotide cytosine, adenine, and guanine is repeated 10 to 35 times. However, sometimes during DNA replication, extra CAG repeats may be added. If more than 36 repeats occurs, then the individual will develop Huntington's disease. CAG trinucleotide codes for the amino acid glutamine. Therefore, the protein produced by the mutant gene has an extra long glutamine tail. These tails cause the Huntington proteins to clump together and disrupt the function of the neurons in the brain. The area of the brain most affected is the basal ganglia, which is responsible for the control of movement, organizing thoughts, and regulating emotions. No one is sure exactly how the mutant protein affects the neurons here, but it ultimately results in them dying off. Typically, symptoms begin to show between the ages of 35 to 50, but in extreme cases it may begin in childhood. Symptoms include involuntary twitching, depression, and memory loss, and patients gradually lose their ability to walk, talk, and swallow. Patients have an expected lifespan of 10 to 20 years after symptoms appear. Death is usually caused by aspiration, pneumonia, infection, or heart failure due to weakened nerve functions.
There is no cure for Huntington's disease. However, symptoms can be managed with physiotherapy and speech and language therapy. The involuntary movement associated with the disease can be treated with tetrabenazine, but this can worsen the psychiatric symptoms. Psychiatric symptoms are treated with antipsychotic drugs, antidepressants, and tranquilizers. Prenatal testing is available for Huntington's disease. Chorionic villus sampling tests for the disease in the very early stages of pregnancy by inserting a needle through the cervix into the chorion and taking a sample. Later on in pregnancy, a sample of amniotic fluid is tested. Lots of research has been done into gene therapy for Huntington's disease. Proteins with neuroprotective effects in the adult brain have been identified. Among them, ciliary neurotrophic factor, CNTF, has been found, which protects striatal neurons. As part of this therapy, a semi-permeable membrane encapsulating a BHK cell line engineered to synthesize CNTF is placed into the right lateral ventricle of the brain. The capsule is exchanged for a new one every 6 months for a period of 2 years.
Created using Powtoon.